Hi, my name is Sathik, and today I'm presenting on canonical spider silk properties through a DNA combinatorial approach. So, why should you care about spider silk? Spider silk actually has a lot of unique characteristics that allow the creation of many new inventions. For example, it's 5 times stronger than steel, but still very lightweight. And this allows for the creation of any boot-proof fabric, such as tough ropes, parachutes, vests, and more. Spider silk is also a strong adhesive. Unlike most glues, which get weaker over time due to water contamination, spider silk actually remains stable. Spider silk is also biocompatible, meaning it's biodegradable and does not usually cause an immune reaction. Due to this, it's possible to create biodegradable plastics to make stuff like bottles. It's also possible to make stronger bandages, stitches, and also synthetic tendons and ligaments, all of which do not cause an immune reaction. Scientists seem to also be creating interesting new inventions every year, such as biological microscopic lenses, as seen in the picture in the right-hand corner, which is only like 2 micrometers long. All right, so spider silk is so amazing. Why don't we see it in our daily lives, and why don't we just make it? First of all, farming spiders is no small task, even if they are physically small. Spiders are not only territorial, they're also cannibalistic. So you can't exactly grow a lot of spiders in close proximity like you can with other farm animals. So the easiest way to generate spider silk is to insert the spider silk genes into other organisms. So far, this has been done in bacteria, yeast, mammalian cells, plants, and insect cells. However, this process is easier said than done because there's no one silk gene, and instead it's composed of many complex repetitive genes. These genes are called spidroids. You can think of them like a gene family that we talked about in class, where an essential conserved housekeeping gene gets duplicated repeatedly. However, unlike genes in the globin family, these genes aren't duplicated a handful of times. These genes are duplicated dozens of times in order to create long chains of spider silk. To help you understand, imagine that each one of these gray circles represents a single gene, each of which contains numerous nucleotides, and each gene codes for a single silk protein. This is very interesting because instead of increasing gene expression through complex methods like we talked about in class, such as activators and inducible genes, evolution instead just duplicate the gene over and over again, so it's going to get read more. However, this is a big problem for scientists, as it's a struggle to just insert one gene, let alone each additional gene being inserted, making the process exponentially hard. As you can imagine, inserting multiple genes in a specific order is practically impossible. Another problem with spider silk is something I kind of lied about. There's no one spider silk that is strong, flexible, and adhesive at the same time. I mean, think about it. Logically speaking, something can't be flexible liquid glue and a rigid bulletproof vest at the same time. It doesn't make sense. However, there are specific spider silks that excel in each quality, and some silks can be somewhat good at multiple qualities. This is possible through different spider gene modules. These modules are genes that code for a specific silk protein, and these give the spider silk a unique property. And the end result is a bunch of different types of spider silk. It follows a similar principle to the globin family that we talked about in class, where both hemoglobin and myoglobin perform a similar function in carrying oxygen, but under different circumstances. For spiders, here are all the different genes that are currently known to cover different silks. As you see boxed in red, even in a single orb-weaver spider web, there are 4 types of spider silks. The MSIP1 gene in the top left corner encodes for a tough silk that wraps around prey. The dragline silk coded by MASP1 and MASP2, which is the strongest silk and the structural foundation of the web. There's flagline silk encoded by flag, which can stretch up to twice the size to capture the prey. And there's PYSP1, which encodes adhesive attachment disc, and it's used to anchor the web and the dragline silk to different substances. The spider egg sacs also contains different silks that are tough, which is encoded by TUS1 and ACSP1. We won't know the difficulties of making spider silk, but there are some benefits from making it. If we understand what gene combinations are associated with different spider silk properties, it could be possible to design spider silk with properties that are better than those found in nature. For example, this paper is trying to create 2 different types of spider silks using the newly discovered spider genes. Library A consists of genes that make up dragline silk and egg case components to make a silk that's tough as possible. Library B consists of genes that make up elastic and adhesive silk to make biodegradable rubber. The way they got these genes for the experiments is that they bought plasmids containing genes of interest for library A and library B separately. As you can see on the picture to the right, they did not keep these plasmids intact. Band restriction enzymes were used to separate each silk modules and give them sticky ends. Now, all these genes need to be inserted into a new plasmid through random head-to-tail litigation of the silk genes within a restriction enzyme site. And all that means is that the beginning piece of DNA module or the head is just an end of another DNA molecule or the tail, and that's assorted randomly. This is so the silk modules are recombinant and new properties of silk can be discovered. This is also much easier to achieve than putting a whole unedited spider silk spideroid into the cell. This should also lead to multiple modules binding to each other, and thus hopefully the recombinant silk link can simulate the replicated long segments in natural silk spideroids. If you look at what's in the red box and image to the right, this is where the BAM2 restriction enzyme site is, and where all the process would be happening, as the same sticky ends that's as the separated modules cut by the same BAM2 enzyme. If we zoom into the BAM2 restriction enzyme site, these are some of the possibilities of what it could look like. Each X in the top diagram represents each silk module circle in the diagram below. If everything goes as planned, the restriction site should contain a bunch of silk modules that are usually not found next to each other in nature, being strung together to sticky ends. The genes should not be overlapping each other and should be assorted randomly, with different genes based on whether it's part of library A or library B sample. You might notice that the green ACSP1 silk module is in both libraries, and that's because it has the stiffness and strength properties that are needed in library A recombinants and has a high elasticity that is needed in library B recombinants, so it's good for both. However, this process is still not perfect, as it's all based on random nature. So getting a transformed colony that's at least 1,000 base pairs of unique combination spider silk genes is extremely rare. After cutting out all the spider silk genes with restriction enzymes and analyzing the data on the gel, they found that out of 652 colonies, only 15 colonies were viable. These spider silk E. coli cells were grown very similarly to how Pligos transformed cells in class, with growth mediums for food, ampicillin antibacterial to kill cells that weren't transformed, and a molecule to activate the silk gene expression. The culture was then centrifuged at 7,500 Gs for 20 minutes, and protein column purification was done with urea buffer, where the targeted protein was eluted at pH 4.5. The proteins were then run on STS page, which is very similar to the gels we run in class, to see the molecular weight of the proteins. The molecular weight of the synthetic silk were still pretty small and weren't really comparable to wild spider silk. So the researchers used directional parameter approach to combine the silk modules of different strains of some of the E. coli. This results in cells labeled A261, A102, and B102217-2, which are the samples boxed in red. And as you can clearly tell, they have a quite bigger molecular weight. The proteins were further purified and freeze-dried. Then sample replicas of the protein were then placed in methanol overnight. Afterwards, the methanol was evaporated. Methanol is known for beta sheet formation in silk-related polypeptides, so a lot of data we'll see will compare silk treated with methanol versus silk that's not treated with methanol. The scientists then performed Fourier transform infrared spectroscopy, which measures all the wavelength peaks of the protein and then correlates that to a specific secondary structure. They chose to focus on these 5 samples out of the 15 because they all had a similar large size and unique domain expressions. For example, A219 is only made up of mass gene dragline silk, and it has beta sheet content of 16.7%. These beta sheets give silk protein their strength. The other recombinant A samples has a dragline master gene and also the genes code for egg case proteins, and thus has much higher beta sheet content. This could be due to the mass genes only encoding for one beta sheet and 2 other secondary structures, while the egg case genes code for numerous beta sheets and in general also just have more secondary structures. With increased beta sheets, these recombinant A libraries with egg case proteins and dragline cells should theoretically be much stronger than natural dragline silk. Slight sequence alterations can drastically alter the secondary structures of the silk, and thus drastically alter the properties of the silk, creating whole new recombinants. For example, B102217 and B102217-2 are similar. They have almost the same names. They have flexible flag protein genes that has GPGXX motifs. And a motif is just a fancy way of saying, say, the genetic code of interest. This motif encodes for beta turn secondary structure. However, B102217-2 has one extra PYSP12 and ACSP1 module, and that's seen boxed in red in the sequence. This means the GPGXX motifs now occupies a smaller percentage of the total protein mass. And this dilutes the effect of the GPGXX motifs, which at the end results in a drastically reduced beta turn percentage and relatively increased overall beta sheet percentage. This could theoretically give these two B library recombinants very different properties. Despite having a similar sequence, the B102217-2 sample could be much tougher but less flexible compared to the B102217 due to the higher, stronger beta sheet content and lower flexible beta turns content. The last data collection method scientists used was atomic force microscopy. As you can expect by the name, this method can yield in microscopic pictures which can be used for topography, which means you can see the surface of the silk. But it can also be used for nanoindentation, which measures how much pressure the microscopic silk can handle. And that can be used to calculate how elastic the silk is and how tough the silk is. The scientists wanted to get good data, so they had some criteria for which samples they wanted these tests on. They wanted high expression of silk gene E. coli because obviously you want to choose the strain that will produce the most amount of silk. They wanted the highest molecular weight silk, so it can be the most similar to natural spider silks, which have a very high molecular weight. They also want the silk with the highest beta sheet percentage, so theoretically it would have the better material properties. And of course, the scientists wanted unique combinations of the original proteins since they're trying to create whole new recombinant silks. And this resulted in 3 samples that we show in the next slides. The image on the right shows the topographic results of these 3 silks. The B10-2217-2 films were smoothed before and after methanol treatment. There wasn't much of a big difference. While both the A261 and A102 had spherical structures before the methanol treatment and a much smoother surface after the methanol treatment. The spherical structures seen in the A261 could be due to air bubbles that are formed when the film is deposited, or it could be due to small aggregates of undissolved protein. And since beta sheets are flat and the methanol treated samples are flat, this also supports the scientist's idea that methanol aids in its formation. This graph shows the nanoindentation results, which measures how strong the silk is in young moduli. The bars in black are methanol-treated samples, while the bars in white are samples that are not expressed in methanol. The higher the young moduli is, the more stress it can take before deformation. The lower the young moduli is, the less force it can handle, and the more elastic it is. The library A samples A261 and A102, while having a lower molecular weight, were still more rigid than B102217-2, which makes sense as they contain more rigid beta sheets from the MASP1 and MASP2 genes, while the B sample contains PYSP1 to fold into random coils. So even though it's heavier, it's still weaker. While it may be hard to tell in B10-2217-2, the methanol does significantly increase the young moduli from 3.4 to 4. However, for A261 and A102, the methanol treatment decreased the young moduli of the films. And the scientists theorized that this could be due to improperly formed beta sheets, intercalating with properly formed beta sheets, and this mismatched design decreased the rigidity of the silk. The young moduli of recombinant silks, which are shown in the black bars, and the natural silk in gray bars, are compared to see if the recombinant silk is actually better. At the top of the graph, A102 and A261 had a higher young moduli than all the native silk proteins. These are good results, as it means that the recombinant A library achieved its goal of containing silks that is stronger than natural silk. However, B10-2217-2 had a lower young moduli than a native silk protein, except for the flag protein, where it has a significantly higher young moduli. And this makes sense as flag protein is very elastic, and B10-2217-2 is also designed to be elastic. However, the B recombinant also has a higher beta sheet content, which leads to being a unique silk that's stronger than the most elastic proteins, but less elastic than the most elastic proteins. It's somewhere in between a strong and an elastic silk, and has a unique property. While this all looks very promising, the young moduli of the recombinant silk was measured on the nanoscale level, while the natural silk proteins were measured on the macroscale level. This is an important distinction to make, as spider silk is known to be stronger when measured on the nanoscale level compared to the macroscale level, so the comparison isn't really one-to-one. However, it's still really impressive that the short 50 to 80 kilodaltons recombinant silk can even compare to the much, much larger natural silk proteins, which can be up to 300 kilodaltons in length. In my opinion, overall, this experiment is a success based on the fact that the researchers created spider silk that has unique properties and better properties than those found in nature. For future directions of this study, they can increase the length of recombinant silk to mimic the length of natural silk. They can increase the types of recombinant silk and they create silks that have unique properties not found in nature. They can also increase the quantity of silk produced, not only for cheap mass production, but because the researchers want to measure macroscale qualities of recombinant silk and compare with the macroscale qualities of natural silk. However, in my opinion, it might be easier to measure nanoscale properties of specific natural silks they want to test and compare to nanoscale measurements of the recombinant silks, since mass production of recombinant silks might take years before it's a possibility. And these are my references. Thank you for listening.